The TANBead 61E-NX reagent enables nucleic acid extraction from whole blood, frozen blood, and buffy coat samples. In this video, we demonstrate the workflow with whole blood. First, carefully remove the aluminum foil seal from the autoplate. Next, shake the blood sample thoroughly to ensure proper mixing. Add 300 microliters of blood sample and 30 microliters of proteinase K into wells number 1 of the autoplate. Insert the spin tips into the Switch 8 instrument and start the tip insertion program. Finally, place the autoplate into the Switch 8 and start the corresponding program to complete the process. In combination with our automated instruments, it efficiently isolates high-quality nucleic acids, ensuring reliable downstream analysis.